गुड मॉर्निंग डियर स्टूडेंट्स, हाउस यू ऑल। सो एज यू नो कि कल हमने जो टॉपिक था रेस्ट्रिक्शन एंजाइम वो स्टार्ट किया था। टुडे वी विल डिस्कस दैट हाउ द रेस्ट्रिक्शन एंजाइम वर्क to cut the DNA, how they recognize the site, what are the types of different kind of restriction enzymes. Okay, so let's start. That restriction enzymes are molecular seizures that have capacity to cut the DNA at specific sites. So in this video, you are able to see that we have a phage, that is a bacteriophage with a kind of spikes that are acting as a end plate. So with this, they have a capacity to incorporate inside the bacterial cell. तो अगर आप DNA को कट नहीं करना चाहते हैं तो add methyl group and the DNA which do not have methyl groups can be cut efficiently with the help of restriction endonucleases. So endonucleases are of two types, as I told you yesterday, restriction endonucleases and exonucleases. Exonuclease remove the nucleotide from the end of DNA, whereas endonuclease make cut only at specific locations within the DNA. Means if it is within DNA, they are endonucleases. Restriction endonuclease always cut DNA molecules at particle point by recognizing a specific sequence of 6 base pair that is known as the recognition sequence. If a DNA base pair have that specific recognition sequence, only then the restriction enzyme can cut. More than 900 restriction enzymes have been isolated from 230 strains of bacteria, each of which recognize different recognition sequence. हमारे पास बहुत से एंजाइम हैं दैट हैव कैपेसिटी टू कट एट स्पेसिफिक टाइप. तो जो फर्स्ट एंड मोस्ट फेमस टाइप ऑफ रेस्ट्रिक्शन एंजाइम है दैट आर EcoR1, वेयर E represents the Escherichia coli, that is Escherichia, that represents the name of genus. The word co, it represents the coli, that is specific name of the genus Escherichia. And the third letters that are written in the italics must be slant, and the name of the strain is R, and the Roman words that are in the last of this word, they represent in which sequence that strain is isolated. Means we have extracted number of strains. How this EcoR1 it works? So we have two sequences. First one is in 5 to 3 direction, other one is in 3 to 5 direction, but both have same sequences. Hence they are known as palindromic sequences because they have same word when read from both forward and backward direction, that is GAATTC. Next, that is GAATTC. So the sequence is same in both the cases. Okay, so the DNA EcoR1, that enzyme, has the capacity to recognize the gene sequence that is GA from the GAATC from the first end, that is 5' to 3' and other 3' to 5' end. The restriction enzyme, it cleave the DNA molecules in two waves. That two methods or two ends are, first one are the blunt ends. If they cut the DNA exactly at opposite point to each other and the DNA are straight ends, means they do not have any kind of cohesive or the attachment type of end. They are also known as even cut ends because they have same exposure sites, means they have a straight line from where the new bases can be added. But the restriction endonuclease generally they cut up in the form of staggered cut. Staggered is the cut then you cut the DNA in the form of helix and they generate cohesive ends. So in E. coli, most enzyme cut two strands in a staggered pattern that leads to formation of the protruding ends, means one end of one strand is longer than the complementary strand of the other strand. These protruding ends generate a cleavage that have maximum capacity to receive the complementary base sequences and hence increases the receptivity or the binding of the DNA. As a result, they readily pair with each other and the ends that formed are known as cohesive end or the sticky ends. These cohesive and have in itself they have capacity to bind with each other with the help of enzyme ligase. The efficacy of binding of sticky ends it increases and the DNA become much more stable as the blunt ends can. So the efficacy, the binding capacity and the ligase action it works much more efficiently in case of the sticky ends, and this sticky end nature is used for the recombinant DNA technology. Restriction endonucleases are used in genetic engineering. As I told you yesterday, that you cut the DNA at specific site with the help of specific restriction enzyme. That restriction enzyme, same restriction enzyme is used for both the plasmid DNA and for the foreign DNA. Both these ends are sealed up with the help of the enzyme DNA ligase that act as a glue. This recombinant vector molecule cannot be created unless the vector and the source DNA is cut within the restriction enzyme. If they are not cut at the specific site, they will not give you the exact sequence. Now, the steps of this restriction enzyme working are the separation and isolation of DNA fragment that can be done with the help of gel electrophoresis. Firstly, you take the DNA, you pour on the cuvettes. As you know, the DNA fragments have negatively charged. Then you switch on the charge. That charge it generates a kind of force for the motion of DNA molecules toward the anode in the matrix of agarose gel. The DNA fragment they move in the agarose gel with the help of their according to their size, and that bands or the color that you observe on the screen is just because of a dye that is known as ethidium bromide. Then you make illusions of that to separate the DNA, and separated DNA is then used for further research. Thank you.